Good morning, everyone, and welcome. I'm pleased to have you here today. We will be discussing sterility testing with a focus on automated systems. I'm Dr. S.K. Singh, and I hold a PhD in this field. In this presentation, we'll explore the latest advancements in automated sterility testing, its benefits, and how it enhances quality and efficiency in laboratories. Let's get started. Welcome to this slide on introduction. Sterility testing is a crucial step in ensuring that pharmaceutical products are free from viable microorganisms. It helps us confirm that the products are safe for use and meet quality standards. Traditionally, these tests have been quite time-consuming, often taking up to 14 days to complete. This lengthy process can delay product release and impact efficiency. However, with advancements in technology, automated systems have emerged as a game-changer. They offer rapid, sensitive, and reliable results, significantly reducing testing times. Their increasing adoption in pharmaceutical quality control labs reflects their importance in modern manufacturing. Overall, these developments help us ensure safety more efficiently and keep up with the demands of the industry. Let's explore the principle of automated systems in this context. These systems detect microbial activity through several key indicators. For example, they monitor CO2 production, which increases as microbes grow and metabolize. They also observe oxygen consumption, since microbes often use oxygen during their metabolic processes. Additionally, some systems utilize fluorescence or color change to signal microbial activity, making detection easier and more visual. Essentially, microbial metabolism produces measurable signals, and automation leverages these signals to identify growth efficiently and accurately. Now, why is automation so crucial in this process? First, it allows for faster product release, which is vital in maintaining a steady supply chain and meeting market demands. Second, it significantly reduces human error, ensuring more consistent and reliable results. Third, automated systems improve sensitivity and reproducibility, meaning they can detect even small changes in microbial activity with high precision. Lastly, automation helps us stay compliant with global regulatory standards, which require rigorous and consistent testing procedures. Overall, integrating automation into microbial detection not only streamlines our workflows, but also enhances accuracy, speed, and compliance, key factors in ensuring quality and safety in our products. Now, let's take a closer look at the different types of automated systems used in our field. First, we have continuous monitoring systems, which are designed to keep an ongoing watch on critical parameters, providing real-time data and alerts. Next, there are CO2 detection systems, often used to monitor microbial growth through changes in carbon dioxide levels. Then, we have ATP bioluminescence systems, which are quite innovative. They detect adenosine triphosphate, or ATP, to quickly assess biological contamination. Lastly, there are pressure or impedance-based systems, which analyze changes in pressure or electrical impedance to identify microbial activity. As an example, I'd like to highlight the BACT/alert system from bioMérieux. This system uses colorimetric detection of CO2, meaning it changes color when microbial growth occurs, providing a clear visual cue. It combines automated incubation and monitoring, streamlining the process from sample to result. This system is widely used in pharmaceutical and clinical laboratories because of its reliability and efficiency. Understanding these different automated systems helps us appreciate how technology enhances accuracy and speed in detecting contamination, which is a critical part of our overall quality assurance efforts. Now, let's take a closer look at how the BACT/alert system works. First, a sample is inoculated into the bottles. If microbes are present, they begin to grow, producing carbon dioxide as a byproduct. This CO2 production is key because the system's sensor detects it, leading to a change in pH when this happens. A color change occurs, which acts as an indicator. Finally, the instrument provides a clear positive or negative signal, telling us whether the sample is contaminated. Moving on to the regulatory basis, It's important to note that this method has been accepted by major authorities like the United States Pharmacopoeia, the European Pharmacopoeia, and the World Health Organization. However, since this is an alternative method, it requires proper validation. That means we must demonstrate that it's equivalent to traditional, compendial methods, ensuring it's reliable and accurate for our purposes. This validation process helps integrate new technologies into established standards, maintaining the integrity of our testing procedures. Now, let's talk about the validation requirements, which are crucial to ensuring our testing methods are reliable and compliant. According to pharmacopoeial guidelines, there are several key parameters we need to focus on. First is accuracy, which means our method must correctly measure what it's supposed to measure without bias. Then, there's precision, ensuring that our results are consistent when repeated under the same conditions. Specificity is also vital. Our method should specifically detect the analyte of interest without interference from other substances. Another important aspect is the limit of detection, or LOD, which tells us the smallest amount of analyte that our method can reliably identify. And finally, we need to perform a comparative study with traditional sterility tests to validate that our new method provides equivalent or better assurance of sterility. Connecting all these points, these validation steps help us build confidence in our testing process, ensuring it's robust, accurate, and in line with regulatory standards, ultimately supporting product safety and quality throughout the entire development pipeline. Let's take a closer look at this example: vaccine sterility testing. The process begins with inoculating the vaccine sample into BAC-T/alert bottles, which are specially designed for detecting microbial contamination. Once the sample is in, the bottles are placed into an automated incubation and monitoring system. This setup allows for continuous, real-time observation without the need for manual checks. One of the key advantages here is the early detection capability, which typically happens within 3 to 7 days. This speed is crucial because it enables faster batch release, meaning that safe, sterile vaccines can reach the market more quickly, improving public health outcomes. This method has broad applications. It's used for sterility testing of various products like injectable drugs, biologics, and vaccines, things where sterility is absolutely critical. It's also applicable to cell and gene therapy products, which are emerging areas in medicine. Additionally, environmental monitoring is another vital application, helping ensure that manufacturing environments remain contamination-free. Overall, this example highlights how automated, rapid testing methods are transforming sterility assurance across multiple high-stakes areas in healthcare and pharmaceuticals. Let's talk about the advantages and challenges of this method. Starting with the advantages, one of the biggest benefits is the rapid results. We're talking about getting outcomes in less than 14 days, which is a significant improvement over traditional methods. This speed allows for quicker decision-making and more efficient workflows. Another key point is continuous monitoring. Unlike periodic checks, this approach provides ongoing data, helping us catch issues as they happen. It also boasts high sensitivity, meaning it can detect even small amounts of microbial activity, which enhances accuracy and reliability. Plus, there's reduced manual intervention involved. This can save time and reduce human error, making the process more streamlined. And importantly, all the data gets recorded digitally, which makes analysis, storage, and sharing much easier and more organized. Now, moving on to the challenges. One major hurdle is the high initial cost. Investing in this technology can be expensive upfront, which might be a concern for some organizations. It also requires trained personnel to operate and interpret the results properly, so some level of expertise is necessary. Method validation is mandatory, meaning we need to thoroughly test and verify the method before it can be reliably used in practice. Lastly, we need to be aware of the possibility of false positives. For example, CO2 detected from non-microbial sources could lead to incorrect conclusions, so understanding the context and limitations is essential. Overall, while this approach offers many advantages that align with our goals for faster, more accurate monitoring. We also need to consider these challenges carefully to ensure effective implementation. Let's take a closer look at how our approach compares with traditional methods. When we talk about traditional methods, we're usually referring to established, often manual or less automated processes. These methods can be effective, but they often come with limitations like slower processing times and higher chances of human error. In contrast, our approach leverages advanced technology to streamline the process, making it faster and more accurate. You'll notice that while traditional methods might require extensive manual input, our system automates many of these steps, reducing workload and increasing efficiency. Another key point is adaptability. Traditional methods may struggle to keep up with changing data or evolving requirements, whereas our approach is designed to be flexible and scalable. This means it can easily adapt to different scenarios and larger datasets without losing performance. Ultimately, the goal is to improve overall outcomes, whether that's in terms of speed, accuracy, or scalability, and to demonstrate how innovative solutions can complement or even surpass conventional techniques. This comparison underscores the value of embracing new technology to meet modern challenges more effectively. So, to wrap things up, let's talk about the future of sterility testing. Automated systems are really shaping that future, offering a lot of exciting possibilities. They significantly improve efficiency, allowing processes to be faster and more streamlined. Plus, they help ensure compliance with strict regulatory standards, which is absolutely critical in our industry. It's important to remember that these systems must meet rigorous validation requirements to be trusted for quality control. When implemented correctly, they become ideal tools for modern pharmaceutical industries, helping us stay ahead in a highly regulated environment. Overall, embracing automation in sterility testing isn't just about keeping up. It's about advancing our capability to deliver safe, effective products more efficiently and reliably. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional information you might need. Just contact us through the provided details, and we'll be happy to assist. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're committed to supporting you on your journey, so don't hesitate to get in touch if you need anything. Thank you once again, and have a great day.